Hello and welcome to IB Biology Notes with Dr. Kimo. This is B1.1 Carbohydrates and Lipids. In this video, we'll learn about carbohydrates and lipids, things like their structure, their properties, their functions, and how they are synthesized. We start with carbon because life is carbon-based. Carbon is essential for all life. And the special thing with carbon is that its atoms can form covalent bonds with 4 other atoms. Some other elements can do that too, but carbon generally forms more stable bonds. Carbon can bind to carbon, but also to many other elements. And this allows for an almost unlimited number of different molecules built around carbon. The atomic number of carbon is 6 and its atomic mass just over 12. The covalent bonds spread evenly around a carbon atom, forming a tetrahedral shape. You can see that on the right. That's the simplest carbon compound, methane. The bonds are evenly distributed around the black carbon atom in the middle. Methane is a potent greenhouse gas. It's the main constituent of natural gas, and it's produced by some archaea as part of their metabolism. In methane, there are no double bonds, but carbon can form double bonds and even triple bonds. Carbon can form long straight or branched chains as well as rings. Most carbon atoms form covalent bonds with carbon, hydrogen, oxygen, nitrogen, sulfur, or phosphorus. And do pay attention to the colors, because carbon is usually depicted as black, hydrogen as white, oxygen red, nitrogen blue, sulfur yellow, and phosphorus orange. All major macromolecules in living organisms, such as carbohydrates, polypeptides, and nucleic acids, are based on carbon. The examples shown here are relatively small carbon compounds. Very large carbon compounds are called macromolecules. There is no strict minimum size limit, but generally you could say more than 10,000 atomic mass units or more than 1,000 atoms. Macromolecules are made up of monomers, and monomers link to form polymers. This is not entirely true for lipids, but we'll talk about lipids later on. You may remember this illustration from a previous video where a nucleotide is added to a growing chain of DNA or RNA. This happens by a condensation reaction where 2 molecules are linked together and water is released. This same reaction is behind the formation of polypeptides, which we'll talk about in another video, and behind the formation of polysaccharides, triglycerides, and phospholipids. Monosaccharides are the structural unit of carbohydrates. Polysaccharides are polymers made of monomers called monosaccharides. Glucose is the most common monosaccharide. There are some things you should know about glucose, such as it's produced in photosynthesis. It is used in cellular respiration, where it releases energy when oxidized. It is water-soluble and very stable. It can disrupt osmotic balance. Osmosis is something you'll learn about in another video, but osmotic balance is very important for cells, and cells cannot store a lot of glucose without disrupting osmotic balance. So instead, they store the glucose as starch or glycogen. On the right, you can see some common monosaccharides. The ones at the top are hexoses. They have 6 carbon atoms. And the ones at the bottom are pentoses. They have 5 carbon atoms. The polysaccharides starch, glycogen, and cellulose are formed by glycosidic bonds between glucose molecules. These bonds are formed through condensation reactions. The hydroxyl group on carbon 1 of one glucose molecule is linked to the hydroxyl group on carbon 4 of another glucose molecule. Whenever there are branches, as there are in amylopectin, which is a part of starch, and in glycogen, these branches form between hydroxyl groups on carbon 1 on one glucose molecule and carbon 6 of another glucose molecule. Polymers can be hydrolyzed back into monomers. Digestion of food includes hydrolysis of, for example, polysaccharides, polypeptides, and nucleic acids. Hydrolysis requires water. And as is the case with all metabolic reactions, hydrolysis is catalyzed by enzymes. For example, your digestive system contains some enzymes which hydrolyze polysaccharides, others that hydrolyze polypeptides, and so on. Your cells then use these monomers, such as nucleotides, amino acids, monosaccharides, to build new polymers or to obtain energy. For example, when you eat protein, your digestive system will break down the proteins into amino acids, and then your cells will build their own protein from those amino acids. Let's have a closer look at the polysaccharides. There's starch, glycogen, and cellulose. And starch and glycogen store energy. Plant cells make starch, and animal cells make glycogen. Glucose units can be removed when energy is needed and added when there's more than enough energy. For example, when you exercise, your muscles need a lot of energy. Glucose is the primary energy source of all your cells, and if there's not enough glucose available in the blood, your muscle cells will start breaking down glycogen to release glucose. Then there's cellulose. It's a structural component in plant cell walls. In starch and glycogen, glucose monomers are linked at an angle, which makes the polymer curved. In cellulose, each glucose is upside down in relation to its neighbors, which makes the polymer straight. You can see this in the illustration on the right. This difference in structure has important consequences, and we will look at the reason behind this difference in structure. Starch and glycogen consist of alpha-D-glucose on the left, and cellulose consists of beta-D-glucose in the middle. The difference between these two molecules is highlighted here. The hydroxyl group on carbon 1 sits at different angles. Imagine that we put two beta-D-glucose molecules adjacent to each other. Then the hydroxyl group on carbon 1 on one molecule and the hydroxyl group on carbon 4 on another molecule are too far apart, and they cannot link together. However, if we turn one of the glucose molecules upside down, then the hydroxyl groups will be close enough and they can link together. This leads to the formation of long cellulose molecules, and they can form multiple hydrogen bonds between them. This leads to the formation of long and strong microfibrils. Carbohydrates also play a role in cell-to-cell recognition. More specifically, cell surface glycoproteins allow animal cells to recognize each other. Glycoproteins are cell membrane proteins that carry a carbohydrate on the outside, and they allow cells to recognize each other which is important in, for example, tissue formation and immunity. One classical example of the role of glycoproteins in immunity comes from the ABO blood type system. If you have, for example, blood type O, you have this carbohydrate attached to one of the proteins on the surface of your red blood cells. Then you can receive blood type O from a donor, but you cannot receive blood type A or blood type B from a donor, because red blood cells of blood type A have a different carbohydrate on their surface, and blood type B another different type of carbohydrate on the surface. If you have blood type A, you can of course receive blood type A blood, and you can also receive blood type O blood, because the carbohydrate associated with blood type O forms part of the carbohydrate associated with blood type A, as you can see from the illustration. The same is true for blood type B. If you have blood type B, you can receive, of course, blood type B blood, but you can also receive blood type O blood, because that carbohydrate associated with blood type O is part of the carbohydrate associated with blood type B. If you have blood type O and you receive blood type A or blood type B blood or AB blood, which is also possible, then your immune system will recognize these red blood cells as something foreign and will try to destroy them. Now let's move on to another group of macromolecules, lipids. Lipids are hydrophobic. They have limited solubility in polar solvents such as water. They are called hydrophobic, although they are not repelled by water. They just dissolve more readily in non-polar solvents like acetone, toluene, and ethanol. Lipids are a diverse group of molecules which includes fats, oils, waxes, and steroids. Fats, as you may have noticed, are solid at room temperature, but they are liquid at body temperature. Oils are liquid at room temperature. Waxes are solid at room temperature and above, but they do melt if you, for example, burn them in a candle. Steroids have a four-ring structure, and examples of steroids are cholesterol, testosterone, and estrogen. Fatty acids are a crucial component of most lipids. They are unbranched carbon chains. Their length is usually 14 to 20 carbon atoms, and they have a carboxyl group at one end, to the left in this illustration, and a methyl group at the other end, to the right in this illustration. They can be saturated or unsaturated. And saturated means that they do not have any double bonds between carbon molecules. They are sort of saturated with hydrogen. If they are monounsaturated, they have one double bond. If they are polyunsaturated, they have more than one double bond. Most naturally occurring unsaturated fatty acids are cis unsaturated, and let's have a look what that means. Oleic acid is a cis monounsaturated fatty acid. It makes up 55 to 80% of olive oil. Lactic acid is a trans monounsaturated fatty acid. It's often found in industrially processed vegetable oils. Now, cis fatty acids have their lone hydrogen atoms on the same side of the double bonded carbon atoms, whereas trans fatty acids have their lone hydrogen atoms on opposite sides. This illustration shows how that looks. You can see cis above with two hydrogen atoms on the same side, and trans below with the hydrogen atoms on opposite sides. What you can also see from this illustration at the top is that cis fatty acids have a bend at the double bond. Hence, they don't pack as densely and they form fewer hydrogen bonds with other fatty acids. This leads to a low melting point. And as you probably know, oils are liquid at room temperature. They have a lot of cis unsaturated fatty acids. Glycerol and 3 fatty acids form a triglyceride. Fats and oils are triglycerides. They are formed by condensation reactions, which is illustrated in the figure below. To the left, you see glycerol and 3 fatty acid chains, and they link together to form triglyceride. And as you may have guessed, water is released. Glycerol, 2 fatty acids, and a phosphate group form a phospholipid. To the right. In animals, triglycerides are important for energy storage. They are stored in adipose cells in adipose tissue. They are chemically very stable and do not dissolve in water. So they are stored as droplets in the cytoplasm and have no osmotic or other effect on the cell. They also have a high energy content, about twice as high as carbohydrates. Triglycerides also play a role in thermal insulation. They are poor conductors of heat. They also play a role in shock absorption. They are liquid at body temperature and they form protective layers around inner organs. You may remember this illustration from a previous video. Phospholipids. They are very similar to triglycerides, but they have only 2 fatty acids instead of 3, and they have a phosphate group. They are amphipathic, and they form bilayers because they are amphipathic. They have a hydrophilic part and a hydrophobic part. Phospholipids naturally assemble into bilayers in polar substances such as water, and the bilayers can then form vesicles. We end this video with steroids. Steroids are lipids with a 4-ring structure. They are mostly hydrocarbon, they are hydrophobic and non-polar. Thanks to these properties, they are able to pass through the phospholipid bilayer. And this is important for steroid hormones whose targets are intracellular. Steroid hormones are synthesized from cholesterol, which is an important component of the cell membrane. The steroid hormones include progesterone, which is involved in the female menstrual cycle, pregnancy, and embryogenesis. Testosterone is the principal male sex hormone and an anabolic steroid. That's all for now. See you next time.